Great.
Okay, I would now like to return to our routine proceedings. I'd like to again welcome all of you to the open session of the National Advisory Council for Human Genome Research. As you can see, I am once again sitting alone in my conference room doing this director's report virtually because of the current pandemic. I look forward to the day where we can all gather again in a room, but for now, these are the circumstances we find ourselves in. I will also tell you, as we've done before, to accommodate a shorter council meeting because of the Zoom format, we've trimmed down my director's report, so it's a little bit briefer than normal. But nonetheless, it contains important collection of updates that I wanted to share with you. And as the same for the rest of the open session, my director's report presentation is being videotaped, and that recording will be made available as a permanent archive on NHGRI's website, genome.gov. For those of you who might be new to council meetings or tuning in for the first time, I want to make you aware of how we have all this organized, a bit like a big electronic resource associated with my director's report, very much like a supplementaries materials on a published paper. You can go to this convenient URL and access this site. All the slides that I will be showing are available to you for downloading, either in PDF form or even in PowerPoint formats. And then meanwhile, we will, as we proceed through the slides, on occasion you will see, or most of the time, you will see a document number in the bottom right corner of the screen. And that document number reflects materials or websites that you can access related to the slide, all nicely organized on this website. And this dedicated website and all the linked documents will be archived on genome.gov as part of a historic record of this council meeting. There's a number of other presentations that will be going on during the open session of this council meeting. I'll try to stay clear of those and let other people discuss those details. Following my director's report, there'll actually be two presentations from working groups of this council. Stephen Joffe, who's the Founders Professor of Medical Ethics and Health Policy at the University of Pennsylvania Perelman School of Medicine, is going to give the annual report from this council's Genomic and Society Working Group. And then following that presentation, Terry Manolio, who is the director of the NHGRI's Division of Genomic Medicine, will give the annual report on this council's Genomic Medicine Working Group. We then have 4 concept clearances that we're going to march right through. Each will be presented by an NHGRI program director. First, Lisa Chadwick will present a concept clearance on supporting talented early career researchers in genomics. Next, Adam Felsenfeld will present a concept clearance on the Centers of Excellence in Genomic Science, or CEGS program. Then Colin Fletcher will step in and he will give a concept clearance on the knockout mouse phenotyping program. And our cleanup batter will be Heather Colley, who will present a concept clearance on curriculum development in genomics, genetics, or genomic informatics for medical students. So those will be the 4 concept clearances that you'll hear later today. Meanwhile, for the rest of my director's report, here are the 7 categories of topics that I'm going to cover. These have worked well for many years, and so we're going to stick with it. And we're going to start with some general NHGRI updates. And the first one is a fun one I'm actually quite proud of. I am delighted to also announce today that literally today we are releasing a new and updated brochure about the Institute. Resulting from an immense amount of effort of numerous staff members, this 35-page brochure nicely showcases the key elements of NHGRI. It's designed for a broad audience, and the brochure features information about our history, our organization, our values, and it also features and highlights key Institute programs from our major research areas. Conveniently, we have made this available both in PDF and flipbook versions on a very convenient website, which you can remember the URL. It's part of genome.gov. Just go to genome.gov/brochure. And you can access and download a PDF version, flip through the flipbook if you'd like. And we have plans to also make printed copies. Those will be available upon request in the near future. So please take a look at this. It is the latest and greatest, and I think it has a very nice way to review many aspects about the Institute in a very accessible form. At the February council meeting, I provided information about our new bold predictions for human genomics by 2030 seminar series. Now, at that time, I announced the first 5 sessions of this 10-session seminar series, and now I'm pleased to share with you the schedule has been rounded out for sessions 6 through 10, which have now been finalized. Now, as a reminder, each of these seminars in the series, we have invited 2 speakers to join us and use a specific bold prediction from our 2020 strategic vision as an aspirational theme for their talks, highlighting their own research within the context of that theme, and then speculating about the next decade in the research area. The sessions are open to the public and are recorded for later viewing on our Genome TV channel of YouTube. For example, the next session will be held on May 25th at 3 o'clock Eastern Time, focusing on bold prediction number 4. Research on human genomics will have moved beyond population descriptors based on historic social constructs such as race. This session is going to feature talks by Charmaine Royal of Duke University and Genevieve Wojcik of Johns Hopkins University. The session is going to be moderated by NHGRI's Ben Spana. So please tune in for these either in real time or watch them after the fact and by looking at the video recordings. Now, speaking of our strategic vision, I thought you'd like to know that under the 21st Century Cures Act, all NIH institutes and centers are actually required to submit a strategic plan to Congress every 5 years in order to keep members updated about any work going on at the NIH. Now, the Policy and Program Analysis Branch is currently drafting NHGRI's required strategic plan for Congress. Now, not surprisingly, this is going to be crafted as a companion document that will go along with our 2020 NHGRI strategic vision recently published in Nature. Relying heavily on that published strategic vision, the Institute's strategic plan for Congress will highlight the Institute's scientific objectives and priorities, as well as to underscore how NHGRI is serving as an efficient and effective steward of public resources. Through these efforts, we'll also develop a framework for crafting the next strategic plan for Congress in 5 years, when there will not be a recently published strategic vision paper to submit at the same time. We wanted to share with you the fact that a number of NHGRI staff members have volunteered to support the Administration for Children and Families, or ACF, which is part of the Department of Health and Human Services, through the Unaccompanied Children's Program at our southern border. As of today, 10 NHGRI staff members have volunteered their time and energy to this effort, and 1 NHGRI commissioned officer has been deployed on a long-term assignment. NIH has well over 200 volunteers who are being deployed to various locations across the United States for 2 to 8 weeks at a time. Each volunteer first travels to a personnel mobilization center in Dallas, Texas initially, and is later sent to another location such as San Diego, California, or San Antonio and Fort Bliss, Texas, Erie, Pennsylvania, Miami, Florida, among other places, with new sites being added across the country as the program continues. Volunteers are providing logistical support, clinical support, and general oversight of the children until they can be placed with their families in the United States. It is a tough assignment, as you can imagine, but it is also extremely rewarding from what we're hearing about those who have been deployed. I am very impressed, although not at all surprised, to see such a giving spirit from the NHGRI staff. Moving on then to some general NIH updates. Starting with our new secretary, Javier Becerra was recently sworn in as the 25th secretary of the Department of Health and Human Services and the first Latino to hold the office in the history of the United States. As secretary, he will focus on ensuring that all Americans have health security and access to health care. Previously, Secretary Becerra served 12 terms in Congress as a member of the U.S. House of Representatives, and then most recently served as Attorney General of the state of California. Now, I actually would note that while a member of the U.S. House of Representatives, he also served on the Smithsonian Board of Regents at the exact time when the NHGRI forged its partnership with the Smithsonian's National Museum of Natural History to create the exhibition Genome Unlocking Life's Code. The board actually had to formally approve the partnership, and so I personally interacted with the then congressman at that time. And I can tell you that he was very excited about genomics in general and the new genomics exhibition in particular, being extremely committed to supporting efforts for educating the general public about important new areas of science and medicine. At NIH last month, Chris Austin departed as director of the National Center for Advancing Translational Sciences, or NCATS. Now, as founding director of NCATS, Chris led the center's efforts to transform translation from an empiric process into a predictive science. Prior to the establishment of NCATS, Chris was senior advisor and director of the NIH Center for Translational Therapeutics, which was hosted by NHGRI. In his new position, Chris will become CEO partner at Flagship Pioneering, a life science platforms company in Cambridge, Massachusetts. Now, in the meantime, Joni Rutter will serve as acting director of NCATS while a national search is conducted for a new NCATS director. In March, NIH Director Francis Collins announced expansive new efforts of NIH to support equity, diversity, and inclusion in science. Organized as a multifaceted initiative called Unite, these efforts aim to address structural racism in biomedical research with the goal of ending racial inequities. The primary goals of the Unite initiative are U, understanding stakeholder experiences through listening and learning. N, new research on health disparities, minority health, and health equity. I, improving the NIH culture and structure for equity, inclusion, and excellence. T, transparency, communication, and accountability with our internal and external stakeholders. And E, extramural research ecosystems, changing policy, culture, and structure to promote workforce diversity. Now, NIH is committed to instituting new ways to support diversity, equity, and inclusion, as well as identifying and dismantling any policies and practices that may harm our workforce and our science. You can expect to be hearing more about the Unite initiative, as well as the programs and policies that emanate from it in the coming months. I would like to remind you that there are several government websites available that provide relevant information about COVID-19, such as those at NIH and at the Centers for Disease Control and Prevention. For NIH grantees and applicants, a COVID-19 resource is available on the NIH Grants and Funding website. In addition, NIH regularly issues news releases regarding COVID-19 related research findings, and NIH Director Francis Collins' blog regularly provides an excellent source of updated information. Now, a recent notice announced the continuation of the temporary extension of eligibility for the NIH K99R00 Pathway to Excellence, Pathway to Independence Award during the COVID-19 pandemic. And a recently released funding opportunity aims to support social, ethical, and behavioral implications research on disparities in COVID-19 testing among underserved and vulnerable populations. Also, Mike Lauer, NIH Deputy Director for Extramural Research, recently posted COVID-19 extramural survey results on his blog, Open Mic. Now, the May Council meeting is typically the time when I can provide an appropriations update that features the first versions of the House and Senate appropriations bills. This year, the President's budget outline was unveiled in mid-April, rather than the more typical February release date. This is the opening gambit, if you will, for the appropriations process that reflects the White House priorities, with the featured numbers not yet having the granularity at the level of individual NIH institutes and centers. A more detailed presidential budget is expected to be released on May 27th. And so this updated budget at that time will include what we anticipate to be more specific NIH institute and center numbers. Well, what do we know so far? What we know so far from the President's budget outline is that the White House is proposing some notable increases. The Department of Health and Human Services is proposed to be funded at $133.7 billion, reflecting a 23% increase over the fiscal year 2021 enacted numbers. The ask for NIH is $51 billion, reflecting a $9 billion increase. Now note, Of that $9 billion increase, $6.5 billion is proposed for a new Advanced Research Projects Agency Health, or ARPA-H, which would be modeled after the Defense Advanced Research Projects Agency, or DARPA, and would aim to drive transformational innovation in health research. That would leave $2.5 billion as the increase for the rest of NIH. Now, of course, all of this reflects the president's proposal, and there will be many twists and turns in the appropriations process until we end up with a final fiscal year 22 appropriation. So stay tuned, put on your seatbelts, and we will do our best to keep you updated. And finally, and in late-breaking NIH news, I'm very excited to tell you that NIH Director Francis Collins recently announced his selection of Zoe the cat as the NIH feline in chief and head NIH zoo bomber. Now, in her role, Zoe will be the top cat for all things requiring feline input, including leading major research programs in the genetics of It's All About Me, and also the genetics of the gastrointestinal consequences of fur balls. Now, in addition, she will regularly break up the important work of the NIH director by bombing his Zoom meetings and making sure that people laugh during what I can assure you sometimes really is a rather boring Zoom gathering. Finally, she will be launching a YouTube channel for cats of NIH staff called the Feline Bombers, which will showcase the best cat Zoom bombing episodes that occurred during NIH meetings throughout the COVID-19 pandemic. Now, Zoe's appointment will become official once she receives all government ethics and security clearances, as well as takes all the required online training, which actually might take so long that it actually happens in the next of her 9 lives. We will, as always, keep you updated about this important appointment as we learn more. Moving on a bit more seriously to some general genomics updates. Starting with a sad one, actually, Mark Bogoski passed away on March 18th, 2021 at the age of 66. Mark was a pioneer in bioinformatics and genomics. He was one of the original medical staff fellows at the National Library of Medicine's National Center for Biotechnology Information, or NCBI. During his career, he served on the faculties of Harvard Medical School, Beth Israel Deaconess Medical Center, Johns Hopkins University School of Medicine, and Fred Hutch Cancer Research Center. He also served as the founding director of the Paul Allen Institute for Brain Science and as vice president of the Novartis Institute for Biomedical Research. Moving on, the National Academies of Sciences recently announced their newly elected members. Of particular relevance to the genomics community and to NHGRI are the individuals listed here. Congratulations to these individuals. And similarly, an impressive duo of genetics and genomics researchers and NHGRI colleagues were recently elected to the American Academy of Arts and Sciences, and we extend our heartfelt congratulations to these colleagues as well. And another honor, Ada Hamish, the Frank V. Sutland Professor of Pediatric Genetics and Clinical Director of the McKusick-Nathans Institute of Genetic Medicine at Johns Hopkins University School of Medicine, has received the 2021 ACMG Foundation for Genetic and Genomic Medicine's David L. Ramoines Lifetime Achievement Award in Medical Genetics. Ada was recognized with this award because of her commitment to teaching and mentoring, her energy and dedication while helping patients and their families, and her leadership in the creation of online resources, most notably as the scientific director of Online Mendelian Inheritance in Man, or OMIM, and co-creator of PhenoDB and Gene Matcher. Congratulations, Ada, and Council Member Hal Dietz. Please do convey our congratulations to Ada for this remarkable honor. We will now move on to NHGRI's Extramural Research Program. Now, NHGRI's Genome Sequencing Program, or GSP, is comprised of the Centers for Mendelian Genomics, the Centers for Common Disease Genomics, and the GSP Analysis Centers. The program aims to use genome sequencing to identify genes and genomic variants underlying human diseases. The program is wrapping up this year, and the centers that make up the GSP are working on their final products and finalizing data submissions in NHGRI's Genomic Data Science Analysis, Visualization, and Informatics Lab Space, or ANVIL. All of the GSP components are working on final papers, resources, and analyses, which will be completed over the next 6 to 12 months. Now, one recent highlight was GSP Analysis Center's 5th annual joint workshop with the National Heart, Lung, and Blood Institute's Transomics and Precision Medicine, or TOPMed program, which took place this past March. This workshop featured talks that describe current GSP and TOPMed data sharing and availability, in addition to talks addressing key aspects of genomic data analysis. 150 plus attendees participated in the joint workshop. Comparative genomics involves using computational analysis tools to compare the genome sequences of different species. The study of a wide range of organisms continues to be instrumental for attaining a comprehensive view of vertebrate genome structure and function. As a complement to the developmental genomic tissue expression project in humans, or GTEx, NHGRI released a notice of intent to publish a funding opportunity announcement, or FOA, for a new non-human primate GTEx project. The major goal of this initiative is to study gene expression patterns in multiple reference tissues across developmental stages in non-human primates and compare them to human gene expression patterns. This opportunity is expected to be published in the coming months with an expected due date in the summer of 2021. The notice shown here contains more details about the planned FOA to allow potential applicants sufficient time to develop meaningful collaborations and responsive projects. NHGRI's technology development program continues to expand and evolve, enabling new and improved technologies to foster genomic discoveries. I'd like to highlight 2 funding opportunities. First, there's the Notice of Special Interest in Advancing Genomic Technology Development for Research and Clinical Application, which applies to the parent R01, R21, and Omnibus Small Business Announcements. This notice encourages a broad expanse of technology development research to enable new lines of research, inquiry, and clinical applications in human genomics. The notice uses standard due dates, with the next due date being this summer. Meanwhile, second, there is a transformative nucleic acid sequencing technology innovation and early development request for application, or RFAs, which include R01, R21, and small business announcements. These opportunities solicit applications to develop early stages of novel technologies that will enable greater than an order of magnitude improvement in one of 2 areas: DNA sequencing and direct sequencing of the diversity of the entire RNA molecules. The first due date for these RFAs is June 25th. The NHGRI Genomic Data Science Analysis, Visualization, and Informatics Lab Space, or ANVIL, is a cloud-based infrastructure and software platform that provides an analysis and computing environment for unrestricted and controlled access of genomic and phenotypic data sets. Now in its 3rd year, ANVIL has expanded its outreach efforts with a focus on training opportunities at both resource-rich institutions and those institutions not traditionally engaged in genomics research. So, for example, in March, the ANVIL outreach team helped to organize the first meeting of the Genomic Data Science Community Data Network. This meeting brought together over 40 researchers, and the focus of the network is to build partnerships from a diverse spectrum of institutions and develop meaningful curricula for undergraduate students. The ANVIL team has also partnered with the NIH Office of Data Science Strategy and Howard University's Virtual Applied Data Science Training Institute, or VATSD, to develop an 8-week, or it's actually a free 8-week virtual training series. Now, VATSD aims to attract and engage underrepresented students and researchers interested in data science applications in biomedical, clinical, and genomics research, with a focus on diseases common to minority populations. The training sessions covered foundational analytical skills needed for research using big data and included an ANVIL-led session on how to use cloud computing to make biomedical data science discoveries. The NHGRI Genomic Data Science Working Group of this council hosted a virtual machine learning and genomics workshop last month. The goal was to define key areas of genomics for machine learning analysis and NHGRI's unique role in machine learning research for both genomic medicine and genome sciences. The event kicked off with 2 keynote presentations and moved on to 4 focus sessions exploring algorithm development, ethical, legal, and social implications, data and resource needs, and clinical applications for machine learning in genomics. The event generated tremendous interest with nearly 1,800 participants from over 50 countries on day 1 and about 1,500 participants on day 2. Video recordings and presentations are now available on the workshop website. The Clinical Genome Resource, or ClinGen, evaluates and disseminates the clinical relevance of genes and genomic variants for use in precision medicine and research. ClinGen's Complex Disease Working Group collaborated with the Polygenic Score Catalog, an open database of polygenic scores and sister catalog to the NHGRI EBI GWAS catalog, to develop and publish a framework for generating and reporting on polygenic scores. Now, the framework includes guidance for score development, including describing study populations, score evaluation, and score translation for clinical applications. And I would point out that 2 of NHGRI's outstanding program analysts were co-leads and co-authors of this paper, as highlighted here. Also, ClinGen and the Decipher Consortia hosted the 5th Curating the Clinical Genome Conference earlier this month, which brought together the clinical genomics and biodata communities to discuss best practices for the clinical use of genomic data, including interpretation and clinical utility, as well as a consensus generation of curated knowledge. Topics included polygenic risk scores across populations, approaches to estimating penetrance, non-invasive prenatal testing, precision oncology, and sharing data and associated clinical phenotypes. The Clinical Sequencing Evidence Generating Research, or CSER program, aims to generate evidence related to the clinical utility of genome sequencing with a major emphasis on participant diversity and engagement. The recent issue of Science magazine that commemorated the 20th anniversary of the giraffe sequence of the human genome included an article from CSER investigator Catherine Phillips. Key points in this article are that precision medicine implementation requires consideration of both affordability and value, and that research on how to best define these terms will continue to be needed. Then in February, CSER investigators led by Barbara Biesecker published an article addressing the need to define the term, quote, usual care, end of quote, in clinical genetic counseling research. Reviewing CSER protocols, published literature on randomized genetic counseling trials, and professional practice guidelines, the researchers proposed a working definition merging elements from existing professional resources, which is summarized in the minimal checklist shown here. And finally, at the American College of Medical Genetics and Genomics Conference, which was held in April, CSER investigators presented 13 posters and 7 presentations. One highlight was a concurrent session in which CSER talks addressed challenges in reporting secondary findings in young patients. The Implementing Genomics in Practice, or IGNITE network, is conducting pragmatic clinical trials to study the implementation of genomic medicine in diverse settings. The first trial, called the Genetic Testing to Understand Renal Disease Disparities Across the U.S., or GARD-US, will investigate whether knowledge of a participant is positive for a high-risk variance in APOL1, a gene associated with chronic kidney disease, results in a change in blood pressure management. As of May 5th, GARD had enrolled over 1,850 of the planned 5,435 African American participants that the study aims to engage, as variants are found almost exclusively in African American populations. A depression and opioid pragmatic trial in pharmacogenomics, or ADOPT-PGX, comprises 3 coordinated trials studying whether pharmacogenomic testing and genotype-guided therapy will improve acute pain, chronic pain, and depressive symptoms in patients who metabolize the relevant medicines at different rates than normal. The study team successfully enrolled the first participant in February of 2021, and as of May 5th, over 50 participants of the planned 4,500 participants total have been enrolled. And lastly, in March, the journal Genetics and Medicine published the Ignite Pragmatic Clinical Trials Network marker paper outlining how the network will generate and share evidence about genomic medicine interventions. The Global Alliance for Genomics and Health, or GA4GH, is an international standards body that aims to accelerate the responsible sharing of clinical and genomic data. The GA4GH marker paper, Genomics and Healthcare GA4GH Looks to 2025, has been submitted as part of a collection of papers to Cell Genomics. In December of 2020, GA4GH was incorporated as a Canadian not-for-profit organization. The legal entity, called GA4GH Inc., will act as the keystone of the alliance, working alongside contributors and funders and host institutions for long-term sustainability. The CEO of GA4GH Inc. is Peter Goodhand of the Ontario Institute for Cancer Research. Ewan Birney of the European Bioinformatics Institute leads the executive committee. In February of 2021, NHGRI co-funded the GA4GH U24 Community Resource Grant to Heidi Rem of the Broad Institute, along with our NIH partners, specifically the National Cancer Institute, the National Heart, Lung, and Blood Institute, the All of Us Research Program, and the NIH Office of Data Science Strategy. And finally, the NIH Cloud Commons is now adopting GA4GH standards for its researcher authentication steps. This past February, the Genomic Medicine Working Group of this council planned and virtually hosted the 13th Genomic Medicine meeting, otherwise referred to as GM13, entitled Developing a Clinical Genomic Informatics Research Agenda. The meeting's goal was to develop a research strategy for using genomic-based clinical informatics tools and resources to improve the detection, treatment, and reporting of genetic disorders in clinical settings. 6 sessions were held, during which experts from government agencies, the private sector, and academia discussed topics related to genomic medicine and clinical informatics. Key recommendations from the meeting included incorporate an implementation component within the overall clinical informatics research framework, advanced research to better understand the interface between human cognition and information technology to improve patient care, and develop research methods to identify and mitigate inherent and pervasive biases in data, information systems, access, knowledge, and clinical algorithms. The outcomes and themes from this meeting will be used to develop a manuscript for publication and to explore potential funding directions and opportunities. Video recordings, the presentations, and additional information from GM13 meeting can be found on the meeting's website. And lastly, in terms of NHGRI Extramural Research Program updates, I want to tell you that to enhance the diversity of the genomics workforce and to strive towards the goals of our action agenda, NHGRI is developing new initiatives to increase the participation of diverse students and faculty members in genomics research. The Institute recently released the F99K00 funding opportunity announcement, or FOA, to support graduate students from diverse backgrounds in their transition to postdoctoral positions, as well as the K18 funding opportunity announcement to support the training of faculty members from diverse backgrounds in genomics. Currently, 2 funding opportunity announcements are also under development. The R01 funding opportunity announcement will allow faculty members to develop independent research, while the GREAT program funding opportunity announcement will provide research experiences to undergraduate students with the aim of attracting such individuals to graduate programs in genomics. Applications for these programs will be funded in fiscal year 2022. So moving on to the NIH Common Fund and other Trans-NIH efforts, we will start with the Undiagnosed Diseases Network. And in February of 2021, the Undiagnosed Diseases Network published two bioinformatics papers. The first, by Cobrin et al., investigated the commonalities in the genome sequencing processing workflows across the 11 UDN clinical sites. It concluded that advances in structural variant detection, non-coding variant interpretation, and integration of additional biomedical data may be especially promising for solving cases that have long eluded a clear diagnosis. The second study by Amiri et al. Developed and evaluated a machine learning model to prioritize evaluation of patients with undiagnosed diseases by retrospective and prospective validation. They found that this predictive model may reduce the processing time for accepted patients by 2/3, from 3.3 months to 1.1 months, by ordering UDN applications based on the likelihood of their acceptance into the program. These bioinformatics efforts contribute towards the overarching UDN model of solving undiagnosed diseases or undiagnosed cases and improving the diagnostic journeys for patients with undiagnosed conditions. The NIH Common Fund Human Biomolecular Atlas Program, or HUBMAP, aims to catalyze development of an open global framework for comprehensively mapping the human body at cellular resolution and to determine how the relationships among cells can affect the health of an individual. HUBMAP recently launched a community effort to identify organ-specific anatomical structures and major cell types and relate them to existing ontologies. HUBMAP is inviting the community to contribute to this effort. HUBMAP has also launched 2 opportunities for undergraduates and early career investigators. The Student Genome Internship Partner Program for undergraduates provides the opportunity for undergraduate students from populations underrepresented in the biomedical sciences to participate in research, mentorship, and training activities within the HUBMAP consortium. The program received over 50 applications, and HUBMAP Labs will host 8 students from institutions across the country for this virtual summer internship. The Jumpstart program for junior investigators program provides junior investigators within HUBMAP an opportunity to undertake scientific projects to help advance their career and develop their independence in the workplace. The first cohort for this program includes investigators from Purdue, Yale, Vanderbilt. HUBMAP intends to continue both programs in the future. And last, in terms of Common Fund initiatives, the NIH Common Fund recently launched the Bridge to Artificial Intelligence, or Bridge to AI program. The Bridge to AI program will propel biomedical research forward by setting the stage for widespread adoption of artificial intelligence, or AI, that tackles complex biomedical challenges beyond human intuition. A key step in this process is generating new flagship data sets and best practices that adhere to the FAIR principles, in other words, findable, accessible, interoperable, and reproducible principles, and critically integrates ethical consideration into preparing data for computation. I can tell you that I serve as one of 5 co-chairs, who are all institute directors, for this Common Fund project. Now, 2 Bridge to AI notices of intent to publish were released last month. The first relates to a planned funding opportunity announcement for data generation projects of the program. And the second relates to a planned funding opportunity announcement for an integration, dissemination, and evaluation center. The anticipated application due date for both opportunities is this coming August, and potential applicants are encouraged to visit the Bridge to AI website for updates and additional information. And so moving on to NHGRI activities in the areas of communication, policy, and education. I can start by telling you that NHGRI extramural grantees and contract recipients sometimes seek to receive offers of support from companies or other third parties, for example, nonprofit organizations or other governmental agencies, for their studies or sub-studies or ancillary studies. And this happens after the grant or contract has been awarded. Now, this type of third-party support, or in other words, third-party involvement, may be valuable for advancing NHGRI-supported research. And in general, NHGRI is open to third-party involvement in NHGRI-supported studies and contracts, so long as the primary purpose of the studies and contracts is not compromised and that all NIH and NHGRI policies are followed. So to that end, NHGRI has developed a guidance for third-party involvement in NHGRI-supported extramural projects, which outlines the type of third-party involvement that NHGRI generally finds appropriate. Effective July 1, grantees and contractors who are considering whether to enter into such an agreement should consult this guidance prior to doing so. If the grantee, contractor, or project governance body is unsure whether the third-party involvement in their research adheres to this guidance, they should contact their respective program director or program directors and submit information about the proposed third-party involvement to NHGRI. The Institute will then review the proposed agreement and provide a recommendation on how best to proceed. Now, providing sufficient and well-structured metadata is a key component of abiding by the FAIR principles for scientific data management and sharing. And FAIR data sets maximize the investment in biomedical research. And so in February, NHGRI published a notice announcing that the Institute will expect investigators generating data that will be shared according to the NIH data sharing policy to be diligent about also sharing the associated metadata. Also in these situations, NHGRI expects comprehensive phenotypic data associated to be shared as well. NHGRI also expects investigators to use standard data collection protocols and survey instruments for capturing data as well as appropriate, as well as to use standardized notation for metadata, that is, controlled vocabularies or ontologies, to enable the harmonization of data sets for secondary research use. Now, these expectations will go into effect on June 5th. Moving on to our communications shop, I am pleased to report that the DC Science Writers Association recently awarded NHGRI science writer Praburna Ganguly the News Brief Award in short journalism in the multimedia category for her work in script writing and producing the NHGRI communications video, The Human Pangenome. Now, the video creatively describes the important work being done by NHGRI through the Telomere to Telomere Consortium and the Human Genome Reference Program. In the association's press release, a judge astutely noted that, quote, explaining DNA is never easy, so I was impressed by their mastery of blending animation and strong writing to effectively tell a story, end of quote. Congratulations to Praburna and the entire communications team for their tremendous work in producing this video. Now, throughout 2021 and 2022, NHGRI will be hosting a Genomics and the Media virtual lecture series featuring trailblazers in science communication. Each distinguished speaker is an expert in communicating genomics across different media, including radio, podcasting, writing, speaking, publishing, and everything in between. The series aims to demonstrate the various approaches for communicating about genomics, as well as the unique challenges and opportunities that each medium can bring. Now, the first event is going to feature Apoorva Mandavilla, who's from the New York Times. That event will take place on May 20th. I encourage you to join, and if you want to register and learn more, please visit the series website. Looking back to April, on April 25th, you may recall that's National DNA Day, and every year NHGRI hosts several programs and offers educational resources to mark the occasion. This year, of course, DNA Day was celebrated virtually. NHGRI did a number of things. First, we hosted 2 H3 Africa DNA presentations featuring 2 H3 Africa researchers shown here. And then on April 23rd, NHGRI hosted Rebecca Johnson, the chief scientist at the Smithsonian's National Museum of Natural History, for the Louise M. Slaughter National DNA Day Lecture. Dr. Johnson shared her DNA-driven science journey, which includes establishing the first accredited wildlife forensic laboratory in the Southern Hemisphere and working on conservation genomics of some of Australia's most iconic wildlife species. And then finally, on May 4th, NHGRI's Kim Jacoby Morris moderated a panel discussion with community leaders entitled Genetic Ancestry Testing, Identity, and the Adoptee Perspective. Lastly, the Inter-Society Coordinating Committee for Practitioner Education in Genomics, or ISCC-PEG, aims to improve practitioner genomic literacy and enhance the practice of genomic medicine through the sharing of educational resources and joint identification of educational needs. ISCC-PEG membership now includes over 200 representatives from societies, professional organizations, NIH institutes, industry, as well as individuals with expertise in medical education. 57 new members joined this year, including 4 new ISCC-PEG scholars who will be mentored by ISCC-PEG members. This year's ISCC PEG annual meeting was held virtually back in February. Almost twice as many members registered for this meeting compared to last year's gathering. And finally, and it will be brief, I'll just give you one highlight, it's a fun one, from NHGRI's Intramural research program. So as I come to the end of my director's report, I just wanted to share this because I just think it's really fun and interesting. So it turns out that a spring episode, actually which took place in March, of the TV show Chicago Med featured the genetic disease Vexus. Now, this genetic disease is a syndrome that was very recently discovered and reported by Dan Kastner's research group within the NHGRI Intramural research program. And in fact, and shown on this slide on the left is the New England Journal of Medicine paper, which was published in late December of 2020. And it described for the very first time this disease Vexus. So one of the storylines in this episode of Chicago Med includes a patient who presents to the emergency room with red ears and a fever. And one of the senior physicians suggests that it could be this newly described disease called Vexus, which was met, I can tell you, by skepticism from the younger members of the medical team. But later, they finally acquiesced and they did genome sequencing. And the skeptics came back and declared, quote unquote, "You're right. Genetic sequencing found that mutation, and it confirmed that the patient did indeed have Vexus." So congratulations to Dan Kastner and his research team, not only for publishing this landmark 2020 New England Journal of Medicine paper reporting the new syndrome Vexus, but also between publishing it in late December of 2020 and having it air in a brand new episode on March 17th of this year in Chicago Med in prime time is really a remarkably short period of time. And so it really is a reflection of the Kastner Lab's work and the attention that it gets broadly, including by Hollywood. And so that's what I wanted to share with you in terms of highlights across the Institute, across the genomics community. Before closing, I want to remind you I can be followed on Twitter at NHGRI_director, where I communicate regularly about NHGRI and genomics to various stakeholders. And also for those of you who want to hear more from me, maybe once a month through an email, please sign up for my monthly email update, The Genomics Landscape, on NHGRI's website, genome.gov, by subscribing under email updates. And of course, big thanks to everybody who helped me put this director's report together, all the slides, all the materials, all the supplementary information, dozens and dozens of people involved. A particular additional thanks to NHGRI communications team and the web team for making the director's report into an electronic resource that others can then access and use. And of course, a special thanks to the ringleader for now over a decade of this effort, preparing my director's report and spearheading the whole effort, Chris Waterstrand, who is shown here after receiving her second dose of the COVID-19 vaccine. And so that concludes my director's report. Thank you for your attention. I will stop sharing and see if any council members have any questions they want to ask me.
So thank you, Eric. Who knew that Hollywood screenwriters read the New England Journal of Medicine?
It is gratifying.
Yes. Any questions for Eric from the director's report?